Okay, good. All right, so super awkward. So I am in Hong Kong. So here's a look for those of you never been here. This is the prettiest. Honestly, I've been here for a few years now, but this is the prettiest thing that we have on campus. If anyone disagrees, let me know. But so this is the oldest building that we have. It's on the old side of campus, but I'm actually housed in the Department of Psychology. We're on the new side. You can see here with these buildings looking significantly different in color. HQ sci students, you should recognize your Centennial campus. So it looks quite, we have two chunks that are quite different on campus. I'm housed in the Department of Psychology, where I teach heavily in our neuroscience program now. It's actually a brand new program that we launched 4 years ago to start offering major minor options in neuroscience for our undergraduates. It's actually not a graduate program. And so in the context of this course, then we've been teaching, I've been teaching the core foundations neuroscience course where I teach some of this content that I will go over today. But myself, in terms of my research, I'm not going to spend too much time on this because it's irrelevant, but I spent most of my time studying this part of the brain. So I'm gonna see if any student here has any idea what the back of the brain there is responsible for. Which sense do I research? Vision. Very good, guys. So our YouTube people are on a bit of a 10-second disadvantage. So you're always going to be beat by our Zoom folks. Vision, exactly. So that's the occipital. That's where I spend most of my time, and I use like a variety of neuroscience-oriented techniques. I'm interested in understanding the mechanisms of basic visual capacities, how we see color, how we see stereo, how we see motion, but more importantly, how in the adult brain we can use certain methods to reorganize the visual cortex, which is, to me, aside from the nerd purpose of understanding, the mechanistic basis of vision, has, of course, a very obvious translational aspect, asking, well, maybe one day we can use some of these basic vision lab-based techniques to reorganize cortex in the visually impaired, right, to help them one day. So I do considerable amount of MRI research. We do some brain stimulation research. But today, we're talking about the motor system, and the motor system is interesting, actually, also for me, because we're not going to talk about this today, but because of its very close relationship with the vision system as well. Hello, TA Blossom. Good to meet you. Great. Everyone take notes. Oh, apparently your TA is taking notes. That's great. But this is my roadmap for today. And one of the awkward things about today is, in fact, that I come here a little bit blind to what you guys know. My assumption is, or what I've learned is that I should be assuming a sort of a senior level secondary knowledge. And obviously HKU side a first year undergrad type of knowledge. And so I expect that for some of you, for all of you, there will be a lot of sort of new terms. And for some of you, it will quickly become overwhelming. So I welcome you to let me know when I've lost you. This happens in my undergrad courses too, because sometimes we assume a lot of you guys. So just stop me and let me know what I need to break down for you and redo. I don't have a lot of slides today, but I tend to elaborate a lot and tell a lot of stories. So feel free, again, if you're not comfortable talking, I don't know if it's set up so that you can speak, but go ahead and feel free and type on both sides. So I plan to give you a walk down of the major components of the central motor system. And I've sort of broken this down for you. The gray matter stuff, which is, everyone knows this gray matter cell body is what drives this gray matter stuff, the neurons, right? And it's most of what we think about when we look at the brain. we think of the brain to be sort of this different color from all the tracts, the axons. And for those of you sort of new to neuroscience, right? Eventually, all our cells need to talk to each other. And our axons, of course, you can think very, very loosely to be those communication cables, although they don't quite, at least not in the complex mammalian brain. Obviously, these cables don't physically attach, at least they very rarely attach from neuron to neuron. But you can think of those cables as these white matter things that I have on these slides. And we'll talk a little bit about the role of these spinal tracts. Eventually, I will touch upon what I call the tiny stuff, which is what a lot of our students really hate, even on HQ side when I teach this, because we're really now talking about how the neurons, once I reach the end of my cable, okay, how that signal gets transmitted to the next neuron. Okay, so we have a bunch of neurons lined up, and one neuron needs to transmit to the next neuron, et cetera, et cetera. And of course, what we're going to learn is this NMJ, which I was a little bit too lazy to write on full. This is the neuromuscular junction. This is one of your learning objectives in your outline there, and that is just a fancy term for saying that interface between my motor neuron that's sending a command to, let's say, my muscle fiber. All right. Eventually, then we'll touch upon some of these popular structures. You've heard about the cerebellum. Most people have heard cerebellum is important for motor commands. We're going to talk about today just a little bit, actually. That's why I put it at the end about its role in motor coordination. All right. So I'm going to start this lecture off by talking about chickens. I've actually never started this lecture off by talking about chickens. I want to ask, in fact, I don't need to ask, I'm sure most of you have heard of this saying here, running around like a chicken with its head cut off. And I wanted to ask if anyone knows about the history of this very, very popular saying, and this very, very common saying. HKU side and Zoom side, please press 1 in chat if you've definitely used or heard this saying before. Thanks, guys. And we're going to have a 10-second delay on YouTube side. Or maybe my HKU students have never used this term. Definitely my lab has used this term because that's me every day. Only Justin uses this term. Perfect. So the question is, where does the saying come from? It's actually from a 14th, 15th century. It dates all the way back there. And what the butchers and the farmers at that time observed was that once they axed off the chicken's head, for whatever reason, the chicken could still keep running for a little bit. And in fact, there was a very, very popular chicken. And this chicken, whoops, I kind of overlapped, but if you can make out the text there, this chicken was called Miracle Mike. And you can actually Google Miracle Mike. This is actually Miracle Mike's picture. And the interesting thing about Miracle Mike was that he was actually, his head was butchered. It was considered a miracle chicken because he apparently survived for 18 months, still kind of flapping and flocking about. The reason why I started my lecture with a chicken is because what I want you to think about is what I want you to get around in your head is that not everything is controlled by solely the brain, right? The thing that is in your head. Because I think one of the unfortunate things about neuroscience, and one of the unfortunate things about teaching neuroscience in university is that a lot of the systems that we can teach while ignoring everything else except for the brain. And certainly I do that in one of my courses as well. And so that's what makes teaching the motor system very exciting, because I'm trying to tell you here that certainly your brain as a human plays a large role in motor control and motor command. But there's another part of the central nervous system that is super important as well. And that second part is the spinal cord. Okay, and so the reason why, let's get back to this chicken for a bit so we can just take off this photo. I'll never come back to the chicken today, so don't worry. And what I want to say is that the reason why the chicken could keep moving was because because its spinal cord is still intact, and in at least the not so human form of a mammal, okay, a lot of their motor commands can be controlled from the brainstem down. Okay, they don't necessarily need the more complex parts of the brain. And so this is sort of where I'm going to start. And I'm going to start with just, again, getting you to realize that there are two main sources of motor control that we're going to talk about today. We're going to talk about the spinal cord, which you can think about as really commands issued by the spinal cord are the commands that ultimately drive muscle contraction. Okay, well, if that's the case, okay, Dorito, what does the brain do? Okay, the brain, you can think of as more of like a master control center. Okay, so it has two functions. It will, preserve the kind of, it will take the spatial information that you have about your world, integrate it with sensory information, and it will help you motor plan. It will help you decide on a course of action. It will also help you, this is the brain, it will also help you decide on the way you're about to execute. Okay, if I want to, let's say, I want to reach for my mug in front of me, how am I going to go about that? And what are the muscles that need to be contracting and stretching in order for me to execute? But that's all that the brain does. Okay, the spinal cord, okay, will take commands from the brain, okay, and issue their own signals to your muscles to eventually drive those contractions. So this is why I wanted to start with the chicken, because I think the spinal cord gets neglected. And so this is, oops, sorry, not moving along yet, but I just kind of wanted to show you that. This is a figure taken out actually out of one of our undergraduate textbooks. You can see here, one of the most simple examples here, you got a bunch of muscle fibers that you need to contract if I want to kind of do this motion here. In order to contract, okay, you can see these kind of cylindrical columns are just cartoon representing kind of the microstructure of these fibers here. And each of these you can see are innervated. I'm going to use this word innervated by specific neural connections that are coming from your spinal cord. Okay, and so the point there is that ultimately, okay, your contractions are driven, the execution part is driven by the spinal cord itself. All right, and I'm going to show you here another figure. Hopefully I haven't quite lost you yet, because what I'm showing you is once I take the spinal cord and I give it a cut, what we call a cross-sectional cut. So imagine, you can see my video there, you guys, on both sides. First of all, what I want to say is remember that your spinal cord is essentially an extension from the brainstem of your brain, right? And so I've got the brain kind of sitting atop here, and then the spinal cord coming all the way down here. What you're looking at in front there is a cross-section. So if I take it on the side and I give it a chop like this, okay, then you're looking at kind of that cross-sectional content. And what I've written here are that those neurons that ultimately send those contraction commands to those muscle fibers are located on the ventral side of your spinal cord. So for those of you not so good on neuroanatomy, dorsal, kind of your back side, okay, and ventral side, your kind of your tummy side, okay, and that's actually where these terminologies come from. So ventral side, kind of on the bottom side, and so neurons here then are the ones that are eventually sending out commands through what we call the ventral root of these spinal nerves out towards your muscle fibers. Okay, I'm going to here further complicate it for you a little bit by showing you that, of course, you can see here there are also other roots on the dorsal side. Okay, the question here is, what are these doing? In fact, these are sensory fibers. These are sensory fibers. So just like we need to send motor commands out through our spinal cord is also this nice interface for receiving data about where our limbs are and the current status of its, contraction state. So it needs also proprioceptive and sensory data coming back in to go back towards cortex, to go back towards the brain in order to give us sort of this feedback of how my motor system is doing. Okay, and so these two then, we've got the ventral roots, which is what we care about today, but then we've got the dorsal roots over here. They will join up together to exit eventually your spinal cord towards your peripheral limbs, right? So you can see that here. It's very hard to sort of maybe understand what these are, but you guys probably have heard about your vertebrates, right? Your vertebral columns. And you can see that you've got these kind of spinal fibers that exit above each of these sections here. I hope I'm not causing anyone to lose their lunch, but I think it's always nice to have a look at the real thing. I don't know if you guys have done bell ringers in your classes, but certainly studying off a cartoon figure is very different from studying off a live specimen. But I wanted to show you here that very much like your cartoon figures, you can see that there are both dorsal and ventral fibers that are clearly leaving here from this spinal cord. And so it is visually pretty accurate here, your cartoons. All right. I'm going to move on. I'm not going to do too much on the spinal cord. I'm going to move on, but this is very important because, again, the spinal cord and the spinal fibers, the spinal nerves that exit your spinal cord are the ones that are innervating and driving your muscle contractions. What I want to show you, of course, is that since your spinal cord comes all the way down from the brainstem all the way to the your tailbone, that means then that we've got a bunch of different exit points that we can identify. And you've heard of maybe your 31 pairs of spinal nerves, okay? And this is sort of what we're referring to here. We've got these, what we call the cervical nerves, which doesn't matter for your purpose, and thoracic nerves, lumbar nerves, et cetera, et cetera. And each of these I want to show you, you can see they kind of go off and innervate muscles of specific locations, right? So the first 8 here, C1 to C8, are sort of your upper body to your kind of upper limbs innervation. And then you've got, for example, you've got your leg ones down here. So it is sort of organized a little bit systematically in terms of the sort of where in the body that these spinal nerves are actually serving. All right, so hopefully that is okay. One of the interesting things about the importance of the spinal cord is, of course, you've probably heard of people who have spinal cord damage. And these individuals, you've heard one of the biggest things is a form of paralysis, depending on where the lesion is along the spinal cord. And now, just given what I have told you already in terms of, you can have an intact brain, okay, but you're certainly, if you have an intact brain, but you can't execute, because that's what your spinal cord commands are doing, the final execution command is coming from your spinal cord, okay, then that's the brain can't overcome that. But one of the biggest things is, of course, research that is going towards trying to offer some solution to fixing these motor deficits. And this is one of the videos that I like showing in my classes. I'm going to play this. I think I remember I fixed the sound here, so both sides should be able to hear this. Just give me a second here. I'm gonna have to mute this.
The objective of our research is to restore voluntary locomotion after severe lesion of the spinal cord. And in this study, what we have been able to achieve is restoring not only voluntary but even adaptive control of locomotion after a lesion of the spinal cord that normally induces complete and permanent paralysis. To transform the circuit below the injury from dormant to highly functional state, we administered a cocktail of pharmacological agents and applied electrical stimulation on the dorsal aspect of the spinal cord. In order to achieve this goal, we developed this new robot that provides support against the direction of gravity, but does not facilitate locomotion in the forward direction. This means that the rat can learn to walk over ground in a very safe environment. And what we observe is that over time, the animal regains the capacity to perform 1-2 steps and then long run, and eventually a paralyzed rat regains the capacity to sprint over ground, climb stairs, even pass obstacles.
Come on, push. Come on. Nice.
The brain establishes new connections. This means that the cut fibers, they regrew, establish relay connection in the spinal cord that enable to pass information from the brain past the injury in order to restore a voluntary control over the circuitry below the injury. The therapeutic impact of this type of intervention in humans is difficult to predict. But this very surprising plasticity and recovery that we have observed in the rat open promising perspective to improve function in humans with spinal cord injury. And we are now investing all our efforts in order to move toward clinical application in a timely manner.
Okay. All right, so hopefully I've driven that message home about the importance of the spinal cord. We're going to come back to the spinal cord a little bit later. And I'm going to introduce to you something a little bit different. That is also very important though. But I'm going to move towards the brain. By the way, since I was talking about the chicken, does anyone know the answer to this? Do birds, which of course a chicken is, have the same brain complexity as a human? Yes or no? No. Okay, good answer. No is the correct answer. The question is, what is it missing? It's actually missing the very traditional 6-layer cortex that humans have. We're going to come back to this in a second here, but I'm going to start with this because what I want to show you, I don't think you can see this. This is my boyfriend, Joe. He's my boyfriend because he never talks back to me. But of course, what we're talking about in terms of cortex is sort of the surface layer of the brain that we're so obsessed about. But what I want to tell you guys, for those of you new to neuroscience, is that this cortex is 6 layers thick. But more importantly, it is evolutionary, rather new. And only rather kind of advanced species have the kind of benefits of this 6-layer cortex. The complexity of the cortex, of course, also increases with evolutionary history. But birds don't have this traditional kind of cortex-like arrangement. It does have your traditional brainstem. It might have structures that we might argue to be analogous to the human cortex, but it certainly doesn't have the same amount of certainly when we're talking about some of these regions today, I'm going to insert regions under each of these chunks here. Our chicken wouldn't have had those. And so let's have a look at what the human cortex can do. And I told you, in terms of execution, we already talked about the role of these spinal circuits, right? So I told you also that at some point I need to make some overall decision of what I want to do. For example, grasp my Starbucks, which is right in front of me and looking very appealing right now. And I'm going to throw a bunch of names at you and then show you in a few slides where they are. Don't get too overwhelmed. Like neuroanatomy is one of these things that it'll take time. And sometimes I forget the name of a particular structure myself if it's not a structure I deal with a lot of times, but I'll show you roughly where they are in a subsequent slide. But these are regions of the cortex, except for this region here is actually some subcortical structures that we'll talk about a little bit later today. But we've got the prefrontal cortex, posterior parietal cortex. Chickens not having the same brain as that of humans. Sorry, Jesse, I can take care of this one. So the point here is that chickens don't have the same 6-layer cortex that humans have. And a lot of our very advanced functions, not only motoric functions, but also cognitive functions, sensory functions, come from having that 6-layer cortex available to us. So chickens don't benefit from that. So we've got overall planning strategy, prefrontal cortex, posterior parietal cortex. We've got then having to select and sequence how I'm going to achieve this overall arcing goal. And that's going to be more of what we think of the traditional motor cortex. No problem, Jesse. We're going to have a look at the primary motor cortex, but also the premotor cortex. And then we're also going to later on today, once you guys are totally lost and exhausted, we'll talk about the cerebellum and the basal ganglia. And then finally, of course, we talked about execution, and that's really the stuff that I was sort of beating into you because I think it's a very underappreciated part of our CNS, the brainstem and also the spinal cord. Okay, so this is sort of how we think the overall hierarchy comes together, and we're gonna visit these parts. I know I started sort of at the bottom, but we're gonna talk a little bit about what the other parts of the cortex do for us. Now I'm gonna throw more terms at you. And I'm going to show you here. It's a nice cartoon brain that has labels, so I won't show you Joe this time. And the way you want to think about this brain is that this is the front. This is your eye side, and then this is the back of your brain. All right. And there are a lot of labels here, but one of the main landmarks that I use to figure out where primary motor cortex is, or where my motor stuff is on the brain, is I first look for what we call here the central sulcus. Okay, and the central sulcus is exactly what it is. Okay, so you've probably heard these terms sulci and gyri, maybe in your previous lectures before this one, right? So the sulci are those grooves, right? These kind of indentations, and the gyri are kind of the not indentations. And anyway, the reason why the central sulcus is important is because the motor area part of the cortex is located just in front of that. Okay, I use the word anterior to that because anterior is, in front, right? So, and you can see here I've circled area 4, area 6. Okay, area 4 and area 6. Now, first of all, okay, before I rant too much about area 4 and area 6, you always hear these numbers, okay? I'm obsessed with vision, so we use V1, V2, V3, but you've heard of S1, S2 for somatosensory mechanisms. Maybe you've heard of A1, A2 for the auditory system, okay? Conveniently, we use M1, M2 for the motor cortex, but there's another labeling convention with quite a bit of history that uses these area number digits, okay? And there are 52 areas, and it was actually done by this guy who was on my very, very title slide. This guy here, Corbinian Brodmann, who was I think he was German, so a neurologist who, in the very infancy of neuroscience research, he decided to look at the histology, which means the structure, the gross structure of the tissue, and try to segregate them into 52 areas, into distinct areas based on both its cellular composition and its actually tissue morphology. And he came up with a total of 52 areas, and he labeled them according to these histological differences. And so area 4 and 6 are these numbers that we use from these, we call them Brodmann areas. So anyway, the point here is we're going to focus on these 2 areas of the cortex. There's area 4 here, which is highlighted in this figure in the pinkish red, okay? And that's what we call today the primary motor cortex, okay? And then there is this bluish area here that we call, or Brodmann labeled area 6. Today, we like to call it the secondary motor cortex. But it is unfortunately for students, when we teach it, it is unfortunately functionally further divided into, you can see kind of this dashed arrow here, into kind of this, what we call the lateral bit and the medial bit of the brain. And so you can see here that not only are they functionally divided, and I will tell you how they're functionally divided in a second, but they're also given sort of different subnames. Okay, so within area 6 alone, we've got what we call a PMA, premotor area, and we've got a supplementary motor area. So, oh my God, all these terms. Okay, why do we care? Why do we care? This is why we care. Oops. I don't know how. I don't actually have the labels right now. Oh, I do. I do have it further down. Okay. So I'm going to show you a little bit further down again. Okay. But basically, it has to do with this proximal and distal, proximal and distal serving. So let's stick to this slide for a second. But I will come back to a little bit more complex explanation of how these divisions work. But for now, again, think of the patch we're concerned about in terms of the cortex, the gray matter stuff, okay, is everything that falls just anterior to the central sulcus line. Well, not everything, right? Because obviously not when we reach the frontal cortex. So if we take a look at first, the immediate part that we today call M1, okay, so just this red patch over here, okay. So I want to kind of show you, this is a bit of an oversimplification, but what I want to show you is M1, okay, much like the sensory patch. You see actually behind the central sulcus is another highlighted region here. It's actually conveniently the S1. And the reason why I like to kind of present them side by side, as you can see here, although one is serving a somatosensory function. So that is, for example, tactile information, okay? And one is serving your motoric output. So that's the theme for today. But I want you to look at what we call this homunculus, okay? This little guy, you might have seen this type of a figure before. And what it's actually showing you, okay, is that as we go, as we outline around this patch of cortex, okay, you can see that there are certain regions that have dedicated parts of the limbs that they're serving, right? You can see here kind of this medial side, okay, to kind of the top here is actually from your kneecap onwards to your feet, right? And as we're going over here, okay, towards a little bit more towards the lateral side now, we've got the hand patch of the cortex, okay? So quite simply, guys, although it is an oversimplification because there's a lot of overlap. So ultimately, if I wanted to send a motor command to move my hand, okay, it originates from the specific patch of M1 over here, okay? And let's say I wanted to move my mouth, okay? And my face lips area, you can see is all the way down to this lateral side. What I'm going to suspect is a lot of you actually have no clue what you're looking at. And this figure relative to this figure. Okay. So I'm just gonna borrow Joe here for a second here. Okay, so again, what you're looking at here, okay, here's Joe. And what I'm gonna do, okay, is take a cut down Joe this way. Okay, so it's basically, again, this coronal cut. Okay, and I'm looking inside his brain from the back to the front. Okay, so you're looking at, again, one of these coronal cross sections. And so as I am showing you, as we're looking from this side to all the way this side, we're actually looking in from the midline of the individual towards the outer part. So from the midline, okay, to the outer part of the brain, to the lateral side. So you see the midline part here is serving the kneecap and the feet, and then the very lateral side, kind of your mouth well, the finer parts of your mouth. Okay, so then here's another question. Why on earth does this doll look so weird? Because we have a disproportionate representation. I'm gonna accidentally give things away here. A disproportionate representation of the face. It looks so tremendously long here compared to, for example, the kneecap. Okay, what I want you to think about is that not every part of your body is given the same amount of treatment in your brain. So some are disproportionately served by more neurons than others. Okay, and that's what this cartoon is sort of showing you. You can see your hands get disproportionately good dedication. I call it in my neuroscience classes, this is the concept of having dedicated real estate to the hand, dedicated real estate to the mouth and the lips area. But this, guys, is also the reason why we have such fine control of our hands, and we have such fine control of how our face muscles move. Okay. In the end, it relates back to how much real estate we ask our brain to dedicate to serving those muscles. Okay, and so the motor cortex has what we call this somatotopic organization, which is just this fancy term for saying it's got this kind of orderly organization for having this patch serves the knee, this patch serves the mouth, this patch serves the hand, kind of thing. This is the idea of somatotopy, and this kind of organization is preserved in other chunks of the brain that have different purposes as well, as I was sort of trying to hint at you. And that includes, for example, the sensory gyrus here. You can see the postcentral gyrus, if we look at it, has very much a somatotopic organization as well. So in that case, if you get some sensation at the kneecap, then that's this region here of the cortex that also gets correspondingly activated. Okay, so that's area 4. Okay, I'm going to come back here now a little bit to this idea of what's contralateral. What's contralateral? Oh, sorry. Yeah, I didn't actually do all the text on my slide. So, so the general story you have right now is that the brain sends commands to the spinal cord, and the spinal cord sends commands to your muscles, right? This is where we are right now. What I haven't really told you, and I'm not going to talk about extensively today, is that at some point when your brain is sending commands down to the spinal cord, the tract that this data travels along, these white matter tracts, it crosses over to the opposite side of your brain. And so for that reason, like your right motor cortex ends up commanding your left side of your body, and your left motor cortex ends up commanding your right side of the body. So that's what this means. And one has a motor homunculus, so that I mean the motor somatotopy, but that this motor homunculus controls the contralateral side. So my right motor homunculus controls my left side, and vice versa. Great, good. Nothing going on YouTube. I think YouTube side fell asleep, which is perfectly normal because a lot of them are my lab people, and they tend to fall asleep in my lab meetings as well. Terrible people. I'm going to come back to the 6-layer organization when we talked about the chicken, and well, what the chicken doesn't have apparently. And again, don't overwhelm yourself, but eventually the commands from my motor cortex, as they are exiting to the spinal cord, need to leave from somewhere. And I was just going to show you that there is a very specific layer that it does leave out of, and that's it leaves out of layer 5. And that's sort of what this whole complex thing. It's just I didn't have time to sort of white out everything to like hide all the other terms. But the point of the slide is to show you that, you know, there's a specific layer that these axons leave to synapse onto your, or to talk to your spinal cord. Okay, and so you can see there, at least from layer 5, YouTube people, to the brainstem and the spinal cord here, and it is noteworthy, although I'm not going to beat upon it, but the neurons that kind of make up this layer 5 are among some of the largest neurons that we actually find in the nervous system. Okay, now if everyone isn't too overwhelmed yet, then we're gonna keep going on here, and we're going to talk about area 6. So again, remember area 4, and we talked about the little motor homunculus guy, right, with the somatotopic representation. But then we also have another patch of cortex that matters as well, right? And that's area 6 over here, this whole blue highlighted thing. And I said it is functionally divided, and it's kind of demarcated here to a lateral part, Okay, lateral part, the side part, and a more medial part, the kind of inner part. And the lateral part here is this, what we call the premotor area. And I'll bring you back those red line notes now. What I wrote here, the reason I say it's functionally divided is because this lateral PMA serves proximal motor units. Okay, more terms. Proximal motor units versus the SMA up here, the medial patch of area 6, which serves distal motor units. It's very simple, guys. It's actually in the term. Okay, proximal motor units, all the things that are closer to the midline of your body. Distal motor units, all the your limbs, your hands. Okay, and so things that are farther from the midline. So my hand is a distal structure that needs to be commanded by a bunch of motor units. So distal motor units are actually controlled by my SMA. Okay, whereas the chunks, maybe my shoulder bit, which is closer to the midline of my body, proximal or to the midline of my body, is driven by this PMA part. Okay, and so that's the main functional division between these two subpatches within this whole blue highlighted area. All right. So area 6 then, together with area 4, okay, then commands everything I just told you, right? I think I'm just gonna, so we don't actually have to go all the way back down here. I think at the very beginning, oops, where is my motor cortex here? This slide over here. So remember again, area 6, area 4, all these things, okay, so confusing, but now you understand sort of where they are in the brain. But again, this idea of making an overarching goal and also having to select and sequence how I'm going to achieve these goals involves very much these two areas. All right. So if I come down here now, okay, if we're okay with areas 4 and 6, there are more patches of the cortex that plays a role here. Just going to fill in the gaps here. And these are the regions that are more important about, you know, sort of the planning of the motion. Okay, and so these aren't your traditional sort of motor areas, okay, because we talked about those. The area 4 and 6 are really your motor, and your premotor areas are really the prime areas we talk about when we think about the motor system. But like everything else, we integrate data from other chunks, right? So the posterior parietal cortex, okay, you can see kind of highlighted over here in this purple bit, but also the prefrontal cortex over here. Okay, and you might have heard, and at least most of you have heard about the functions of the prefrontal cortex, at least, right? For these sort of decision-making processes, okay, for really evaluating what we call, a lot of the things that we call executive decision-making is done over here, right? And so it's no surprise to you that the motor system integrates stuff from your executive areas in the frontal lobe. And it should also be no surprise that your motor system also needs to integrate information from what we call the posterior parietal cortex, because I think I said at some point, in order to plan my movement, I need to have data about where my limbs are currently located in space, right? Certainly me trying to grab my Starbucks mug over here is going to require a much different path from when I am currently sitting like this than, for example, when I am currently having a resting posture of having my arm, whatever reason, behind my head. Okay, and so these spatial relationships, okay, and having to integrate all other kind of your sensory data from your other systems as well, then becomes also quite important. All right. Okay, so we still good on both sides. I'm very busy here because I'm staring at chat from both of you guys. Now, what I want to do is give you an example of some of the research that goes into what we tell you. You know, it's very interesting. I'm sure this happens to you in your other classes too, right? Your prof just tells you the facts and you just have to take their word for it. But of course, the facts we tell you have research behind it. I want to show you one of these cute studies. I'm going to focus on area 6. Okay, again, for those of you, and I don't blame you for already forgetting where that is. Again, this blue chunk here. Jesse, we have a question over there. Can we approximate these cortex stuff as the functional areas of the cerebrum? They are certainly part of the functional areas of the cerebrum, but there are other functional parts, right? Functional just means serving a function, quite frankly. So these, yes, but also everything highlighted here. There are undoubtedly, you've probably read, junk kind of casual articles that I've told you, actually they're not junk, that have told you that we don't use a significant proportion of our brain. That is true. That includes the cortex. There are parts of the cortex that don't really have dedicated functions to them. And so if that's your question in terms of, can we classify these as functional parts of the cortex? Oh, absolutely, absolutely. These are one of some of the key functional parts of your cortex. I forgot what I was saying. Area 6, and I was going to show you the data. So yeah, area 6, remember, okay, area 6 here, bringing it back all the way to this slide here. Motor and premotor cortex, these tactics of how I'm going to go and execute this action, okay? And so how do I know, for example, that area 6 is specific to planning the tactics, and not specific to execution, and not specific to the overarching executive planning and strategy making of your motor command? How do we know that area 6 specifically is in charge of how I'm going to move my muscles to reach that mug? The tactics. So I'm going to bring you back to this monkey slide I wanted to talk about, which is a slide I teach in my 2101, guys. And so this is called the ready, set, go task. So basically the monkey is trained to press one of 4 buttons in front of them, depending on which one lights up. Okay, now imagine that what I'm actually doing as the monkey is performing this task is I'm actually recording from a specific area. I'm recording over this patch of area 6, which again, I just taught you is specific for serving the function of tactic making, okay? How I'm going to execute this reaching command. And so what you're gonna look at here is the activity kind of in a schematic way of a PMA neuron. It's just a short form for an area 6 neuron. And the way to understand this is more firing of this area 6 neuron is represented by more of these vertical lines. Okay, so so here what you're looking at is basically not a lot of firing, a lot of firing, and then kind of reduced firing. Now let me just give you some more context here. So in the ready, set, go paradigm, this is what's going to happen. In the ready phase, the monkey has basically been given all the task instruction and knows, what to do given the rules. Okay, so it knows in terms of like executive wise, the rules of the game, right? But because this is at this point of my ready, set, go paradigm, all we've done to the monkey is, okay, there are no lights on right now. Okay, so the monkey right now shouldn't be engaging in its area 6, right? Because it hasn't needed yet to make a decision about where to press. Therefore, it hasn't needed to make a decision about the tactics for how to press that button. Okay, and so under the ready situation, my PMA, my area 6 neurons, are totally not interested. And you can see that when we do some electrophysiology and we stab that, okay, we don't really stab, but we record activity out of these PMA neurons, and you see that they don't fire at all during the ready stage. Okay, well now let's see what happens during the set stage. At the set stage, the light comes on one of these 4 chunks, okay? And when the light comes on, okay, now what's the monkey thinking? The monkey has decided, okay, where to press, and it should have at this point decided how it's going to press it. The tactic planning should have happened, okay, at the set point. There's no execution yet because because there's actually a trigger signal for the monkey that he must wait for before he must execute. But look at what happens here now to my area 6 neuron. You can see the firing increases quite a bit in this area 6 neuron. Okay, and so this is sort of a sign of, okay, the light has come on, okay, and the monkey has made his decision and also planned how he was going to execute this motor command. This tactic selection has happened, and that's why this PMA neuron is firing like crazy. Okay, then we have the go command. The go command, of course, at this point, we're beyond the rules of the game. We're beyond where to push and how to push it. We're saying spinal cord go, right? And so at this point, then you see once the trigger stimulus comes on and he's good to execute, you see that the PMA neuron again then reduces its activity, right? You can see how just by this task alone and the different aspects of this task, we can have a good idea as to what the PMA is actually interested in, which is in this kind of this set stage of motor tactic planning. Okay, we all good? I don't see problems on either side. Okay, good. So at this point, the PMA neurons stop being interested. Great. All right. Now, guys, don't cry. Don't cry on me. If you've seen YouTube Cry Style, definitely I've seen that in some of my other classes. But here is where we are. We said so far, I'm going to keep recapping for you so you don't get lost. Okay. My cortex stuff, the gray matter stuff of the brain, okay, is responsible for understanding the rules of the game, but also eventually responsible for the planning and the tactics of how I'm going to issue that command. At some point, once I've done the tactics, I've got to issue the go command. Let's go do it, body, right? And that go command travels down from the brain to the spinal cord, where the spinal cord, then you saw those 31 divisions of the spinal cord have nerves that exit it to control these muscles to go execute command. What I want to go to for a second here to visit, because this is actually going to come back to my chicken story. It's all about the chickens today. I hope we all eat chickens tonight, or maybe not. Is that I want to think about as we are descending from the brain to the spinal cord this time. I'm going to draw a picture, and my HKU side students know that I can't draw, so I'm very sorry for you guys. I don't know if you can see this. Please tell me if you can see this. Okay. As I'm going downwards here to the spinal cord, I know my YouTube side can see this. I don't know if my Zoom side can see this. I've sort of given up. Okay, great. This is my best impression of a body. So you can imagine how the rest of my classes go usually. So I meant to represent the brain here. It's obviously not circular, but let's leave that alone for today, shall we? And we've got the spinal cord coming down. And then I said, eventually we've got these spinal nerves that need to exit to my muscle. Okay, I want to fix our terminology problem here. And in fact, I think HKU side will have this terminology problem as well. I want to make it all clear to you. The fibers that are exiting from the spinal column towards your muscles, okay, are called nerves. Okay, I know it sounds trivial, but because I'm going to be talking about tracts, Now I want you to think about why students get confused. Because both tracts and nerves are white matter highways, okay, that send signals down, okay. But how we usually classify it in neuroscience is that we call nerves the ones that exist outside of the central nervous system, okay. And so anything that is not encased in bone, this is how I teach it so you can remember it. It's not encased in bone because it exits out those spinal gaps, right, and towards your muscles. Those are nerves. Those are spinal nerves. What I want to talk about now, and we talked about those way back in the beginning of this class, what I want to talk about are the spinal tracts. So I'm talking about the stuff still inside your CNS and the stuff that is coming now from area 4 and 6 in your cortex to the spinal cord. Okay, so remember, I've got to come this way before I'm going to send commands outwards through the nerves. Okay, those are tracts. Okay, these are spinal tracts. So I'm talking about between the brain and going to the spinal cord at this point. Okay. And what I want to tell you is that there are actually two main categories of tracts that we need to care about. Okay. As we're going from area 4 and 6 to the spinal cord. Okay. The first category, okay, are what we call the lateral pathways. Okay. And the lateral pathways are usually what we think about, actually. If you're a motor system person, you almost exclusively care about this because these are the ones that really come from the cortex itself. Okay. And these ones control dominantly, at least in the human, any of your voluntary movements. Okay. And we have a second category of pathways. Okay. These ones you see in my red line notes are brainstem controlled pathways. So in fact, they don't quite originate up in, the gray matter area 4 and 6 on the surface there, but they originate a little bit further down. I'm going to get rid of that because that was very distracting there. Because I need to show you this picture for those of you who, neuroanatomy problems, right? So again, assume area 4 and 6 somewhere on the surface here, right? Okay, and so we've got our main category of signals traveling down through our lateral pathways, but we've also got secondary tracts, okay, that originate from the brainstem itself. And the brainstem here is further down, okay? You can see midbrain, pons, medulla, okay? That's your brainstem here, quite deep into your brain. These ones are in charge of, things like posture and some of your reflexes as well. Okay, so for us today, and really to prevent you from getting totally overwhelmed, let's focus on the lateral tracts because I need to get back to my chicken story, right? And the lateral tracts here, okay. If you just look over here, it's just showing you again, if we took a cut through the spinal cord and just have a look at where they travel. And that's why this kind of rather crappy figure is not that informative. But you can see here, there are two clusters of tracts here. There's one, this rather fat one here, called the corticospinal tract. And then there's kind of a smaller in diameter cluster of fibers called the rubrospinal tract. And what I'm going to tell you is that, well, we can actually find out. What I'm going to tell you is both of these lateral tracts more or less serve the same function, okay, which is direct this voluntary control of movement from the brain, okay? So the question is, why do we have both of them? And what I'm going to say is that it seems to be an evolutionary story here. It seems to be that we first evolved like other non-complex, that's subjective, but not as complex species. For example, like the chicken, we may have first observed a rubrospinal control from the brain, okay? And it was only much later as humans developed this complexity of having a 6-layer cortex, having the luxury of having this cortex, that then we later developed this lateral, this corticospinal part of the tract. Okay, and the reason we think that it has an evolutionary reason for existing is based on from what we see from lesion data. So have a look here in my text. Unfortunately, I seldom like to do this, but it's all text, this slide. Let's say we cut both available tracts that serve voluntary movement control. So I'm going to lesion both the corticospinal and the rubrospinal tract, okay? What we're going to have, not surprisingly, okay, is that we're going to have fine postural control and fine reflexes. Why do we have fine posture and fine reflexes? Because I told you that that's not served by the lateral pathways, right? That's served by the ventral medial pathways that we're not going to care about today, okay? We're going to have quite a bit of deficit in having an independent control of different fingers, different muscles, because of these lesions. But what I want to say is, look what happens if we lesion only the corticospinal tract. Okay, I lesion only corticospinal tract. I still have one more lateral pathway, the rubrospinal tract, right? And what we're going to have initially in such a patient is that they will have deficits that are very similar initially to the above here, but you will see an eventual recovery of motor function. Okay, the point here is that eventually what we see is that the rubrospinal tract can actually take over function from the corticospinal tract as well. Okay, and so what we have in the human in terms of the lateral pathway, so again, the tract that is connecting the brain and then sending to the spinal cord before we send out to the muscles, right? What we have is a little bit of a very nice redundancy, okay, that is evolutionarily built in, okay? And of course, what the chicken doesn't have is very much going to be this type of luxury, because it doesn't lack the same, it doesn't have the same complexity as us up in cortex. All right. Okay, good. How are we doing over here? checking in to everybody. Still alive apparently for now. Good. A little bit difficult, right? But I think maybe not. Even if you get lost in the terms, the premise is still the same. Okay. Gray matter stuff, the cortex, okay, the planning, the decision making, and then down to the tactic making. Okay. But eventually the execution, okay, needs to come down to the spinal cord, right? My cortex needs to send information to my spinal cord via these spinal tracts, okay, for my spinal nerves now to go talk to my peripheral muscles. Okay, that's what I spend an hour talking about, honestly, and chickens. All right. All right, now, one of your other learning objectives was the this tiny stuff here, your neuromuscular junction. So now here's what we're thinking. Eventually, okay, as I'm exiting my spinal cord through my nerves, the sizes of both tracts doesn't, we have a good question, Jesse. So the size of both tracts doesn't really play any role in maybe one having a higher function than the other, right? I think, so the sizes of the tracts, more broadly speaking, not just the motor system, has an implication for the speed of transmission. So what do you think? A fatter diameter tract is going to transmit faster, or a smaller diameter tract? This is a little bit of a difficult question, actually. Just take a wild guess. This is, I don't know if I have any, I do have some 2101 students here. Yeah, exactly. A larger one is going to transmit faster. Good. And for those of you who can't quite get your head around this, we don't need to get into the details. But the way I like to teach this is think about like small wires as having greater resistance than big ones, because you're a little bit more spacious, right? So your ions, okay, try not to talk about ions today, but signals travel faster. The larger one, yeah. So not just the motor system, but across all your sensory systems as well. The fatter one will give you more reliable and faster transmission. In that sense, then, Jesse, I wouldn't say it has a higher function, but it certainly gives us a better reliable transmission. Small stuff. So hopefully that answers your question. So signal. Oh my God, I have a writing problem. When the signal is traveling, oh no, I don't. When the signal traveling down the spinal tract eventually reaches the muscle, it needs to talk to the muscle. Okay. So this is going to garner a lot of hate on HKU side. They really hate to talk about synapses. And I'm going to try to just, I'm going to try to make this easy for you. I don't know if it's even entirely possible. I don't know, again, because I didn't pay attention to the schedule. I'm not sure if you already had a thing on synapses in your lecture series. But I've got a very complex picture for telling you that eventually my motor neuron that is sending a signal out to my muscle needs to talk to my muscle cell. Contract muscle. And when we talk in neuroscience language, Okay, it's called synaptic transmission. Synaptic transmission. So we call this a synapse, right? Basically, just a bunch of pictures on how a synapse might look like, kind of. TA, Kayla. Sorry to step backwards. Didn't quite get why more complex cortex mean we should benefit from another pathway. Talking. Yes, no problem. We benefit in the sense that we have a fail-safe. We benefit in the sense like we just talked about. We benefit in the sense that we also get probably a faster and more reliable transmission in that sense. But we also benefit, and what I really meant by benefit in that context was because then we benefit from more finely controlled motor commands. Because what I didn't actually tell you is that out of these two lateral tracts, that one of them truly originate from areas 4 and 6, and the other one actually doesn't quite. The other one actually originates from the midbrain. So it's not as privy. It does eventually get data from 4 and 6 and other cortical areas, but indirectly. So you can think of, if we want to just take it back to the chicken example, I think more simply put is that the chicken doesn't benefit from the additional executive elements that humans benefit from given these additional corticospinal tracts. Great. Oh, synapses. Yeah, okay. Guys, we're not going to do too much of the synapses, although I do have a number of figures on these. Eventually, there are actually some more cool stuff I want to talk about. So, and I'm starting to take a little bit longer than I thought. But the first question is, first of all, we talked about how those white matter tracts, whether we're talking about the spinal tract between the brain and going to the spinal cord, or white matter tracts exiting the spinal cord to my muscles, okay? There are signals that are going down these information highways. What you're lacking, and I'm not sure actually if you have had a lecture on something that covered this, is what are the actual signals that are traveling down these information highways? And I again, I don't want to go into too much about this because it's removed from, this isn't a basic, sensory transmission or signal transmission part of neuroscience class. So I don't want to do too much of it, but you do need to know, okay, that eventually this stuff is being carried by proteins, right? What you've probably heard, neurotransmitters, right? And what I want to show you here is just some pictures and some just general logic. So if you miss some specific words and terms, like try not to get too worried. I'm gonna try to help you sum up what happens here. The neuromuscular junction is just a specific name for the synapse that is happening, the communication interface that is happening between your motor neuron coming out, sending information from your spinal cord, okay? I think my computer just froze. Oops, I think we had some technical problems. Do we have is it TA around? Hey, Kayla, I'm having problems sharing my screen. It says disabled screen sharing.
Oh, I can make you the host. 1 second.
Sorry, I think something happened after I crashed here.
Okay, I think you can now.
All right. Oh yeah, I'm good again. Sorry about that. Everything just crashed. And actually, I don't quite remember where I was before we lost everything. Synapses, probably this slide here. I think we had a question though. Humans have a 6-layer cortex. What does 6-layer mean? Navina, did you get that and that thing answered? Yeah, basically, you see this surface of this Joe here. I'm trying to see if I can hold it up to the camera. And you see in fact, what you're looking at, it feels like only sort of 1 layer of gray matter, but in fact, there are 6 layers, okay, of cortex. And I think you can't really see this because he's not exactly gray on the outside. But if you were to sort of slice cross-sectionally, then you would actually see 6 layers of it, of that gray matter. And each layer, although again, beyond the scope of today, each layer will have its own specific, usually very specific subset of neuron types that do specific things. So for example, in these sensory systems, layer 4 likes to receive things. Okay, back to NMJ. Okay, right. So basically, where everything is, yeah, sorry about that, guys. I honestly had to reboot and come back on because everything was just crashing up. Right, so the interface where the motor neuron is talking to your muscle, and that's all you have to think about in terms of what that fancy term refers to. But what I want to say is that the NMJ is historically very important because it was one of the key interfaces that neuroscientists studied to try to understand just general mechanisms for how neurons spoke to each other. And so again, have a look here at this figure. Eventually, my nerve coming out of the spinal cord needs to synapse on to my muscle cell, okay, in order for it to talk to the fiber in order to contract. All right. So again, don't be too worried if there are like too many terms here, but I just want to give you kind of an overflow about how, like I mentioned, the first question I had for you was what actually are these signals? And I said these signals actually proteins that are these proteins called neurotransmitters that are like sort of carrying data down and between cell to cell. And in general, okay, you guys actually might have learned this already in some of your secondary work, but in general, we kind of go through these stages, right? We have a synthesis part inside the cell, okay, that needs to, you've probably heard of these terms, the DNA and the RNA, right? And these RNA chunks need to be replicated, okay? They get sent outside your cell nucleus, and at some point you need to be able to have protein synthesis based on this kind of, these RNA, DNA instructions. eventually, okay, all these neurotransmitters and any other proteins actually gets packaged down and sent to their respective locations. In the case of neurotransmitters, okay, very often they will get sent to the end of your axonal terminal. Okay, you guys are familiar with those terms, right? Like I think I don't need to go over those, right? The cell bodies and the axons and the axon terminals. You guys should be pretty okay with those, right? But eventually, the core of having a cell-to-cell speaking to each other is the release of these neurotransmitters onto sort of a little synaptic gap, and then the uptake of these little proteins, these little neurotransmitters on your second cell. So cell one over here and cell 2 over here, right? And so the NMJ specifically will have its own specific neurotransmitters that are working here. And so I'm just going to add a lot of red line notes here for you to kind of turn this very generic textbook figure into an NMJ synapse. Okay, again, the synapse is that interface where we have cell talking to cell. Okay, so whether it is we have our spinal cord nerve cell talking to our muscle fiber cell. Okay, let's take this as an example. Okay, what's happening here? So we've got our spinal cord presynaptic nerve cell here. Okay, trying to talk to our muscle fiber down over here. Okay, and so you've probably learned the principles of how how electrical transmission works, right? So we're going to have a action potential that is going to arrive, okay, in your first cell, and that's going to trigger the release of these protein packages, acetylcholine. These are your neurotransmitters over here. Okay, I'm gonna give you a second to jot that down for. Oops, sorry. There we go. Stop pressing buttons. There we go. Acetylcholine are your neurotransmitters here. That's happening at the NMJ. Remember that my second cell, which is going to be my muscle fiber here, needs to uptake these signal packages, these neurotransmitters, right? In order for that signal to be transmitted. Okay, and that's going to take the form of these acetylcholine ACH receptors. Okay, and so these ACH receptors are going to bind these acetylcholine packages. Okay, and as they bind these acetylcholine packages, okay, again, not sure if you learned this, again, do ask if you're totally lost, but the binding is going to trigger, okay, a change in the overall potential, overall positivity, let's say, of the second cell. And that's going to cause final consequence here, okay, the change in the internal positivity of the voltage, okay, inside that second cell, quite simply speaking, okay, will drive calcium release, which then triggers muscle contraction. That's sort of as simple as I could make it for you today. But for those of you who don't even care about the names and don't even care about what is happening here, all you're thinking about is this, okay? This signal information is being carried by these protein packages, okay? And at some point, these protein packages need to make it from cell 1 to cell 2, okay? My nerve cell fiber coming out of my spinal cord to my muscle fiber in order for me to contract. In order to do so, okay, specifically the NMJ will have its very specific names for its neurotransmitters and very specific receptors on the fiber side, okay? But it is the rebinding, or it is this kind of transmission of these neurotransmitter packages that will lead to a net change in the internal electric potential of your second cell, right? And that is itself is going to change or is going to trigger a calcium release, which then is going to trigger muscle contraction Believe me, this slide was a lot more complicated, but this is sort of as simple as the explanation as I could make it. Okay, so again, hopefully that doesn't it doesn't overwhelm you too much, but like this is the NMJ. It's just a very specific synaptic junction referring to the motor junction here between the spinal nerve cell and the muscle fiber. And we're involving very specific neurotransmitters here, acetylcholine receptors, and then calcium in order to trigger the muscle contraction. Okay, but it works. If you study synapses under any other context in the nervous system, it works the same way. Okay, it works the same way. All right, good, great. All right, now, actually I think this is more fun, but it's actually not any more easy. So I'm going to conclude with a little thing about a structure that you might not think matters, but it matters a lot. And I want to give you a different perspective in terms of how you think you initiate movement. Okay, because everyone thinks we initiate movement by, we're going to excite, the cortex, then we're going to excite the spinal cord, and then we're going to excite, the nerve cell, and we're going to do another excitation to tell a muscle to move. Okay, the story is actually not quite like this. All right, and so we've got some other problems under different contexts here that I'm going to introduce these structures here, the basal ganglia and the cerebellum here. So here's the premise here. So we now know how movements are planned and executed, cortex, spinal cord. Okay, but there are some missing pieces. Okay, again, I feel like I'm a little bit overwhelming you with these anatomical labels, but again, don't be too worried, because I'm going to go over the general gist of why we care about the basal ganglia. Okay, the basal ganglia is not cortex. Okay, it's not quite cortex, but it's a set of subcortical nuclei. And I think we had a question again about the 6-layer cortex. If we look very carefully, if you have dissected a brain, okay, which we actually do in our first foundations course here, what you will see, okay, is that you will see kind of this pinkish area, which is actually what we call the gray matter. This is the cortex around the outer side. And then you will see that contrasted with what looks like a lot of fatty tissue. That's your white matter stuff. Those are all the axons that we're talking about. Okay. And when I was talking about the 6 layers, and I kept talking about it, although this looks like to you from the naked eye a single layer, in fact, there's 6 layers here going down. Okay. And so that's what I kept talking about there. The point here is the basal ganglia are all these things in the green. All right. And all these nuclei in the green collectively, okay, matter quite a bit when we're talking about initiating action. And I'm going to try to work with the story here without totally losing you or myself from a technical standpoint here. Okay, so we've got something called the striatum. Doesn't matter if you don't remember it, but they're labeled here. Okay, we've got something called the globus pallidus, which is a little bit kind of inferior here, a little bit deeper. Okay, these 2 chunks labeled here. And then we've got something called the substantia nigra, even deeper over here. All these are, subtle nuclei that make up this cluster of thing called the basal ganglia. And again, it's subcortical because it is not a part of cortex. It's subcortex, okay? That's what we mean by subcortical. Now, the biggest thing, what we're going to learn is one of these nuclei constitutes a major source of output to the thalamus. And it occurs to me, of course, that most of you don't know what the thalamus is, okay? But it's a very key region of the brain. Again, it's a subcortical region that likes to feed back up to cortex, okay? So if you want to think about why we care at the moment for the basal ganglia, you can think about these cluster of nuclei as constituting a part of a feedback loop towards cortex, okay, to constantly update and initiate movement and tell the cortex, okay, we need to stop, we need to inhibit, we need to stop movement, stop stretching, stop contracting this fiber, Okay, so we need something that goes back up to cortex, and the basal ganglia plays a very large role in this. Specifically, the globus pallidus plays a large role in this as well. Just more cartoons. I'm actually going to probably skip this so you don't get too overwhelmed. But since you're recording it, you can come back. But I wanted to give you a cartoon for like how these green spots correspond to sort of how you might look at it from a lateral perspective. But I'm going to skip over this at this time. This is the point I was just making here, and it needs to be absolutely clear because then I'm going to show you a animated GIF that is basically the highlight of this lecture if the chickens weren't. Okay, so again, the basal ganglia, okay, receives extensive output itself from the cortex, but it also feeds back into cortex. And I kind of write that all down for you here in the corner. So the cortex in particular, those executive and the kind of decision-making regions and the spatial regions here feed into the basal ganglia, which then feeds into the thalamus, which feeds back to cortex to, to keep it online in terms of giving it a current status of, do I keep stretching? Do I not? Should I inhibit and stop stretching my muscles? And so the flow is something like this, okay? And what I want to say, so I've just put a little note here. Usually what we're feeding back up to is area 6, which we talked about a little bit earlier. What I want to talk about is why we care so much about the basal ganglia, and specifically the substructure of the basal ganglia called the globus pallidus, is because of its role as a motion gatekeeper. Okay, a motion gatekeeper. And you can see in my little notes that I've written down here, okay, what it actually does as a structure is it by default keeps our motor programs and motor actions turned off. Okay, if you think of yourself, I'm going to bring the GIF on now, and I'm going to come back to this slide. Okay, if you think of yourself as, if I had no GP, if I had no globus pallidus, okay, I would be doing something like this. Okay, so you natively want to turn on your motoric actions, but you are tonically inhibited because of the existence of your globus pallidus. That means in order for you to move, you need to release actions from their inhibition. Okay, so I'm going to say this one more time. You are by default wanting to be like that animated GIF. But because of your basal ganglia and how it loops with your thalamus and back to your cortex, you by default keep a pretty good handle on your motoric programs by keeping them turned off, by keeping them inhibited. By extension, then, the way you actually want to understand motoric execution is we do it by releasing the action from its inhibition. Okay, does that make sense? Okay, so my O1 students should have already learned this. Okay, and that's why the GP, the globus pallidus of the basal ganglia, is called a gatekeeper. Okay, because you can see here, it provides tonic inhibition to the thalamus, holding back the flow of possible responses. And when I want to move, okay, what we're going to do, okay, if all these were all possible responses and my basal ganglia keeps them all inhibited, then that means when I want to do something, I need to disinhibit one of these canals. So selectively removing tonic inhibition releases one particular response, okay? And this disinhibition then occurs via this striatum to globus pallidus type of connection, okay? So hopefully that is plenty clear. It's very, very important aspect of the motor system that you do need to keep in mind. I think I have eventually, I do have one slide. I think you guys should have it as well that kind of writes this out. And it's not that complex at all if you have understood what I just said in words. Okay, so again, okay, all motor actions are inhibited by default. Okay, selection and initiation of a particular action then involves disinhibiting the corresponding thalamic neurons. Okay, so again, at rest, okay, the globus pallidus exerting tonic inhibition on the thalamus, holding back all your flailing around. And when you want to action select and action execute, okay, you are inhibiting, okay, this globus pallidus, which then disinhibits the thalamus in order for you to act. Okay, so this isn't something I expect you to remember now or really have a very good handle on, but the premise is you need to disinhibit the inhibition in order for your motor program to proceed. And that's how we act. Okay, it's not just about turning things on. Okay. In this case, in our motor system, we work a little bit backwards. It's about turning off the inhibition, okay, in order to move. Okay, and I think that's one of the cool things actually about the motor system. All right. Okay, I'm going to skip this as well because it's actually more of the same. But I want to now give you two food for thoughts. I'm nearing, like, I think I really will finish on time, so don't worry. But I want to talk about two very, very common diseases. One is neurodegenerative, one is also genetics related, and you guys will definitely have heard of it. Parkinson's and Huntington's disease, and how they relate, and why I taught you about the basal ganglia and about this disinhibiting the inhibition thing in order to move, because now you are totally equipped to understand what is happening in Parkinson's and Huntington's. We're just going to give you a slice of the basal ganglia here. Again, this is another structure here. This is not the globus pallidus, but this is another subnucleus that was highlighted a few slides ago. This is the substantia nigra. And the substantia nigra, actually, if you see here, actually, you can see it's kind of darkish in color, and that's because it contains quite a high level of the pigment melanin. The point here is that the substantia nigra contains a particular subtype of neurons that produces another type of protein, dopamine, okay? And dopamine is critical for your motor system because when we were talking about releasing actions from their inhibition, dopamine is key to doing this, okay? Dopamine is key to disinhibiting the inhibition of your thalamus on your motor programs, okay? So that, by extension, means if I had a lack of dopamine, okay, then I should have a lack of motor control. All right, we still all good? Or some of you guys are already asleep, not entirely sure. Which brings me to the first of the two I want to talk about. And then I'm going to give you one slide on the cerebellum and send you all the way. Parkinson's. I usually, when I'm in class, ask my students what they think the most characteristic symptom of Parkinson's is. And what I hear usually is the tremors, right? Indeed, I do understand why you know, that is probably the most salient thing that you see of a Parkinson's patient, these kind of almost like repetitive actions. It is a mini tremor that you see. But I think the tremor gives you a false understanding of what is actually happening here. Parkinson's is not defined by excessive movement, okay? That is not why they are tremoring. What's actually happening there? Parkinson's is primarily characterized by what we call here hypokinesia, okay? This is a paucity of movement, which means basically a lack or rigid or just not good movement, slowness of movement, rigid movement, okay? And given what I just told you between about the relationship of just now one of the nuclei of the basal ganglia, okay, the substantia nigra, it being dopamine producing, right, which now we understand to be those key protein packages, key neurotransmitter packages that drive the disinhibition of action or action inhibition. Given that we know this, okay, now look at this picture that I have in front of you and look at the SN of a diseased brain, a Parkinson's brain, versus an SN of a totally normal brain. okay? And what you see here, obviously, is much reduced black pigment, right? In fact, we have a degeneration of these dopamine-generating neurons in a Parkinson's substantia nigra, okay? Which means, okay, it becomes very difficult to initiate and sustain motor actions, okay? It becomes, remember this whole thing, I kind of zipped around here, okay? Dopamine is key to driving this disinhibition in order to act. If my substantia nigra is damaged, okay, I have insufficient dopamine, okay, to drive this disinhibition. And so motor execution becomes quite difficult. Motor initiation and sustaining a motor command becomes very, very difficult, okay? That's the key thing about Parkinson's, okay? Now, giving another image, and then I'm gonna do Huntington's and then the cerebellum, and that's it. All right. Again, now these are more images now this time, not so cartoonish, and I want you to understand how to read sort of this type of an MR image, okay? Now, all you have to do, it gave you kind of an arrow to mark where the substantia nigra is in this MRI image. So now we have a healthy control adult here, okay? You can be looking at the SN about here. I lost my highlighter pointer after my crash. And then here, look at a Parkinson's patient and their SN here. Okay, and you see this significant dropout in signal. So it actually reflects a significant drop in tissue volume here in the Parkinson's patient. Okay, and so again, this is the reason why it's a lack of dopamine generation problem. And so it should be no surprise to you then that one of the things in terms of pharmaceuticals that a Parkinson's patient might be given to help relieve some of their motoric problems is something that might promote the presence of dopamine in their nervous system. Okay, so there are something specific. So you guys might not have heard of it, but there are dopamine precursors, L-DOPA, that we could provide these patients with that would sort of enhance the dopamine content there. Okay, so this is the Parkinson's problem. Oops. The second problem, Huntington's, is what you guys might actually have thought was Parkinson's. This is the excessive movement problem. Okay, this one is a genetics, heavily genetics problem. Okay, so Huntington's here is characterized by a lot of abnormal movements, very often accompanied by, I wrote here, dementia and personality and mood disorders as well. And the reason is actually due to where the degeneration happens in a Huntington's patients. You see, actually, it's a lot of degeneration here, specifically in the basal ganglia. Of course, you're going to say, oh, well, Parkinson's was a degeneration as well, but it was specifically in the substantia nigra, but also sometimes in the midbrain as well. But here, okay, because the basal ganglia, in addition to serving your movement loop back to cortex, is also actually serving a lot of cognitive functions, including memory functions. So if we have a rather broad gray matter loss in the basal ganglia, it should be no surprise that in addition to movement problems, we're going to also have these other cognitive so dementia-like problems and memory and mood issues as well. All right, so I'll show you here this time, and then we'll get to the words in a second. Tourette's, so the question here, sorry, YouTube. I hope you guys are still commanded. Yeah, so they're still connected. Yes, Sumed says, is Tourette syndrome also related to dysfunction in the basal ganglia? You know what? In fact, this one has some. So I'm not sure if you guys over on the other side are actually hearing me, because I'm seeing like everyone drop out there. Yeah, I'm not sure. Tourette's, what I understand is actually more than the basal ganglia. It does involve the basal ganglia, but I think it's the entire loop of the cortical striatal thalamo back to cortex pathway. So I have to look into it though specifically, but it's not isolated to the basal ganglia as I understand the Tourette's to be. Okay, Justin says you guys are working. Okay, I don't know. Yeah, because my side had you guys frozen, but I think Zoom is fine. Great. So Huntington's, right? Have a look here. Again, extensive destruction of the basal ganglia, and you can see that's why, compared to the healthy control, okay, you can see why the ventricles look particularly enlarged here, because there's quite a large loss of neurons here around the basal ganglia structures. Okay, and here, remember I said Huntington's is different from Parkinson's because it's characterized by excessive movement. Now let's try to understand this. What's happening here, okay, remember the globus pallidus, which is where I started with the flailing animated GIF, right? That is the thing, okay, that is severely damaged in Huntington's. And that means that there is a decreased inhibition. Remember that tonic inhibition that the GP provides to the thalamus in order to keep all your motor programs in check. Okay, in this particular case in Huntington's, that tonic inhibition gets destroyed. And so in this case, this is what we're talking about if you're thinking about a loss of control of movement resulting in this, you know, excessive flailing. I hate to call it flailing, but, you know, unintended excessive movement. That is Huntington's, and that is specifically because of your loss of tonic inhibition. And that is exactly why it was important. Although I know it was a little bit tedious for you to learn so much so quickly, it's been very important for you to understand how ultimately actions get released. Actions don't get stimulated. Actions get released from their tonic inhibition. And I think when you think of it in the context of what happens in Parkinson's, when you have the lack of dopamine and therefore, you know, the lack of the ability to disinhibit, right? In the case of Huntington's, you have a severe degradation of the globus pallidus, and therefore you have an ability to have this tonic control, the tonic inhibition of your motor patterns. And when you think of it in the context of these two diseases, I think everything becomes a little bit more clear as to why the basal ganglia are very key to your motor loop. Although, as I said, they also play key roles in your other aspects of cognition and memory. So that's why I insisted on kind of throwing that into your lecture today. More MRI images. I'm almost done my lecture. And so again, you see the enlarged ventricles in a patient with Huntington's disease because of the degradation of the basal ganglia. Again, look at this. Your control has a healthy caudate, healthy putamen. These are some of the terms I threw at you when I introduced all the subparts of the basal ganglia. Imagine now, in addition to that, of course, you have the GP over here, which is the inferior part after the putamen. That's also destroyed. And so then, as a consequence, you look at this kind of rather enlarged ventricle in the case of the Huntington's. And because of all that, because of the very key role of the globus pallidus in maintaining that tonic inhibition, you get that excessive rhythmic motion pattern in your Huntington's disease patients. I had originally a couple of short clips that I wanted to play, but I'm going to run out of time. But what I'll do is I'll send, I think I was talking with Alden, so I'll send her the links so that it can get forwarded on, because I don't think I have the TA's emails, or one of you guys can email me after that. One missing piece that I didn't teach in your LOs, but is thankfully rather simple, is of course the cerebellum, right? And this is my sole slide for the cerebellum because everyone wants to go home now. The cerebellum, it's really funny because in Chinese, so I was educated in Canada and I learned all my stuff in English. Now learning the cerebellum in Chinese is called the small brain. Is that how we understand it in English too? So it's this kind of really complex, very highly kind of striated substructure of your brain. We've all heard about how the cerebellum is important. I want to fill in why it's important just very, very briefly here. In the context of motor coordination, I think what we haven't quite talked about, I talked about how, we could have the motor planning from the cortex and the tactic planning, and then everything gets sent to the spinal cord. Eventually, spinal cord sends nerves out to the muscles. But what we talked about stuff, great, thanks, Suman. What we haven't talked about is there's got to be other checks installed into our central nervous system in order for us to keep ourselves coordinated, right? Although, yeah, the cortex plays a good role in planning and stuff, but at some point, we need to be able to coordinate all these different motor programs as well. And that's where the cerebellum plays a role in. I just gave you the text here. Adjust timing sequencing of different muscle contractions, sorting out these multi-joint movements, how they should be executed, what should be stimulated in what order. That's what the cerebellum is key for. And it's interesting because we learned a great deal from patients with lesions in these key structures, right? Just like we learned a great deal from checking out what happens in the Parkinson's patients with severe SN degradation, in Huntington's patients with severe globus pallidus degradation. If we look at patients with cerebellar lesions, okay, what we find are very uncoordinated actions, very inaccurate movements as well, because they don't benefit from this timing and sequencing ability. And I'm just going to, I think I have an image, and then again, MRI image here where we circle a healthy cerebellum and an unhealthy cerebellum. And you can see here, okay, the healthy one is pretty, full, lots of gray matter, lots of white matter. And you contrast that with just like the sheer content that you see in the same brain on the left side, okay? And you can see how, visually you can already see the difference between the two. And so on the left-hand side, this individual would likely be, at least compared to the individual on the right-hand side, this individual would be much more likely to be uncoordinated and inaccurate in terms of their final motor execution. All right, and so I'm going to conclude with a summary. I think I don't traditionally include the words in my summary when I send out my slides. And so here's my summary. And I think, firstly, I get it, a lot of terms, and but I hope the general gist of the story you have been able to capture today. Okay, so motor control involves just about everything, right? And it involves certainly what you classically think, the cortex, the brain, okay, the gray matter stuff. And we went through the key patches of the cortex that are relevant here, area 4, area 6, right? Area 4 for finally the selection and the tactic making, but then it also includes other cortical areas beyond 4 and 6 that feed in, right? Parietal frontal cortex feeding in, for example, my spatial coordinates and integrating other data from other sensory cortices. But the thing is, always remember the chicken. Okay, the brain is just half the story. Okay, that stuff needs to reach the spinal cord because ultimately the contraction, okay, of your muscles, okay, is commanded by your spinal cord. Okay, and so we talked about, again, the distinction between the various nerves that exit the different spinal columns, right? And the key difference there was that depending on where they exit, they serve muscles of different parts of our body. Okay, that was a simple story there. We talked a little bit about the neuromuscular junction, right? Because eventually, gosh, my nerve fiber coming from my spinal cord needs to talk to the muscles, right? And the way, like the rest of the nervous system, the way we talk to other cells is by sending via these biochemical, pouches, right? These neurotransmitter proteins, releasing it from the first cell to the second cell, and then to the third cell, right? From the second to the third, and from the third to the fourth, like this, right? So the principles of synaptic transmission are very much the same across all sensory and motor systems. Just what's different is that I'm working with acetylcholine. I'm working with acetylcholine receptors at the level of the muscle fibers. In between, I added twists to you that weren't quite in your learning objectives, but I think are equally as important. We spent a little bit of time in the second half talking about the basal ganglia, these subcortical nuclei, okay, that you learned are key to prevent yourself from being that animated GIF, right? Because the motor system doesn't work in terms of initiating by, just let's now orderly excite everything and excite my arm muscle, okay? Motor system is better thought of as something that is tonically inhibited, all your motor programs, and that when I need something, I selectively disinhibit. Okay, and so we learned all these things, and we learned about the relationship, and we learned about the logic of why Parkinson's is like Parkinson's and why Huntington's results in this excessive movement pattern. Okay, and then finally, we talked about how cerebellum, in that one slide, I hate not to do it justice, but we just don't have time, is key for keeping everything coordinated, all the muscles in what order, so that's key as well, okay? And so then my final kind of thing was, remember I was talking about, obviously we have, remember that from the brain down to your spinal columns, we're traveling down these spinal tracts, and we had these two major clusters, right? We had the voluntary control lateral tracts, which in the human we've got redundancy in, and then we've got the ventromedial tracts, which serves our kind of reflexive postural control. Okay, so hopefully, so that's all I have for today. Brought right to 6 o'clock Hong Kong time. I apologize for the earlier crash. It was honestly hard crash. Part of that I think is probably like I hear the server going behind me, and I'm gonna blame it on the guys in my lab who sabotaged us.
I would like to thank Professor Dorita for delivering this amazing lecture. Thank you so much. And it was you kept the enthusiasm high, and I think that energized everyone, and we were all listening throughout the field, and it was really a great talk. And you managed to put in so much of information, and yet you delivered everything, like give a broad overview of the entire topic, and I believe you did an amazing job. Thank you so much.
Thanks, Sasha. Thanks.
Yeah. Do you have time to take some questions?
Yeah, I do. I'm going to turn you guys up because technical problems abound.
Okay, if there's any question we have for Professor Dorita, please raise your hand and or just unmute yourself. Yeah.
Okay, if you raise your hand, I'm not going to see it, but the tutors will flag you.
Professor, I have one question. There are you spoke about the lateral tracts that were going, and so there was this the cerebellar the one that is going from the cortical region is thicker than the one that's going from the mid to the rubrospinal tract.
Yeah, that's fine.
Kyla and I were having a discussion on that. So she raised up the question that why does it needs to be thicker? What could be the reason why it's thicker than the rubrospinal tract?
So what could be the reason? I think we mentioned this a little bit. I think this was this right before my crash or right after my crash? I think one of the, it is truly thicker. And one of the things that I brought up was, like the rest of the CNS, that the thicker gives us a more reliable transmission. And a faster transmission as well. Right. So I think from that perspective, so why? In this case, we are a little bit, this is an evolutionary question. I like to believe that humans, because of the increasingly complex behavior requirements that we have to do, right? That we have then gradually evolved kind of a more efficient. So I mentioned out of these two lateral tracts that one of them was newer, right? This cortical spinal is newer. And so it from an evolutionary perspective, it makes a lot of sense for us to sort of look at the cerebrospinal tract and say, you're okay, but we can do better. And so it feels like over history, humans gradually evolved this larger tract to both be able to serve and integrate all these different cortical functions. Again, we talked about how we've got cortex, but the lower mammalians don't. So on the one hand, we get direct cortical input by developing this new tract over evolutionary history. On the other hand, we get faster transmission and more reliable transmission by developing the secondary tract as well. So, it could be, and I'm sure, from an evolutionary perspective, that could already be enough reason for us to have developed these things.
Right. Thank you. Thank you.
Certainly, we didn't develop it as a fail-safe, right? I think we shouldn't think of it like this. Although I did present it as, well, it's nice because we've still got the rubro, right? And it's interesting. It's a little bit curious because, to my knowledge, although someone can kind of prove me wrong, again, I'm a vision person, but to my knowledge, the cerebrospinal tract doesn't give us anything in addition that the corticospinal one doesn't do. So the question is, why did we preserve it? And I think that's a very interesting question to have.
Yeah. So is it like to make sure that there are multiple checks mid-brain from the cerebrospinal tract being preserved?
Sorry, I couldn't quite make it out. Is it to make sure?
I'm saying, is it to make sure that there are multiple checks, and so there's this one check in the lower level, and there's this other bigger check in the higher levels. So could it be something like that?
So in the normal healthy human, although I'm saying the cerebrospinal tract doesn't give us anything in addition that is unique to it, that the corticospinal tract gives us already, in the normal human, both tracts actually still function. And so is it so that it gives us a fail-safe? Again, like I sort of doubt that's why we evolved it. But yeah, I'm not sure how to, honestly, I'm not sure how to speculate on why we have it. Because certainly if we were talking about the development of the human nervous system, it doesn't quite make a lot of sense for us to develop a secondary pathway that we don't quite need, right? And in terms of evolutionary history, we usually develop and prune things that are quite specific to what the human needs.
Thank you.
I guess what I'm trying to say is redundancy isn't usually built into the CNS.
Okay. And do we have an idea of what happens if specifically the cerebrospinal tract gets damaged and the corticospinal tract is intact?
So I just lost you at the end there. So is the question, what happens if the cerebrospinal tract is damaged but not the corticospinal one, right? Yeah. In that case, then you're fine.
Okay. Thank you.
Yep.
I don't see. I think you were very good with the lecture, so you made sure that all the questions are answered. So I don't see any hands here or anyone unmuting themselves. So I don't think there are too many questions they have. I think
okay, great. Yeah, I don't see anything on YouTube side, but that's because I think they all went to dinner already. Yeah.
Sumit has this question on the chat. If you can see.
The striatum is inhibited when GP releases an action to the thalamus, whereas, I see. Okay, so I'm gonna bring back I apologize for skipping. I lost a few minutes because of the crash, but here it is actually. Okay, so not exactly. So the order is a little bit different, is Sumit. So when we want to release an action, what's actually happening is the striatum gets excited. And when we excite the striatum then will inhibit the GP. And inhibiting the GP is the only way we can disinhibit the thalamus like this. Whereas it is not inhibited when the GP is inhibited. I don't know if that helps you, Sumit. So yeah, a little bit backwards in logic there. So it's the excitation of the striatum needs to happen in order for us to inhibit the GP, in order for us to disinhibit motion. Now, let's tie this back to the dopamine problem in Parkinson's again. remember when I said Parkinson's patients, the lack of the dopamine is the reason why they can't release action very well. And that's why you get this rigidity. In fact, this is exactly what dopamine is exciting. Dopamine is responsible for exciting the striatum in order to start this whole chain of disinhibition. Okay, that's how it ties together. So hopefully that helps you with that question.
Yeah, I think that answers it.
Perfect.
Yeah.
Great, Sumi. Yeah, it's, you know what, I think it's, I think the motor system is cute. I think it doesn't get enough attention. And there's so many little things, you know, that are so curious about it, because, you know, if you were to walk in and you were to ask a student, I think you would always think, you wouldn't think about this animated GIF, right? That's not how we would think, you know, it should work. So in fact, it's really cute that, you know, we got to keep ourselves tonically chill. And we got to disinhibit our programs.
And I think everything that we do, humans is all about movement. Like from
Yeah.
talking to anything, it's all about motion. So yeah.
So what I'd like you guys to remember, there's a lot of details of it today, but remember, of course, the motor system, like all the other systems of the CNS, don't operate in isolation, right? So always remember, at the same time, of course, it needs to integrate information from the other senses. Of course, it needs to integrate information from, you know, proprioception about my current body position, right? So there's quite a bit. There's quite a bit to integrate, and that's why we need that, you know, the cortex back to the spinal cord, back to the cortex, back to the spinal cord. We need all these loops all the time, because we need to keep ourselves updated. Do I keep stretching? Do I, do I contract? What is the current status of my body? And so it's very important. Like it's when I teach other sensory systems, it's the same thing. It's very important to remember that system doesn't work in isolation, but it is still rather cute when we look at it in isolation. Okay, we have a couple things here. We have a question here from Jesse, and Jesse says, what weakens Broca's area, and if it gets weakened, does it affect Wernicke's area? Are you guys actually, did you guys have a lecture on Broca's and Wernicke's? I'm not sure if I can, I'm gonna try to find a picture here to help us.
We had a talk on the sensory systems, so yeah, a little on that. In the last lecture, it was a little covered.
I'm just going to answer this question by giving you a Google image. because it's very hard to talk about the areas without an image. Oh God, these images are terrible on oh my God. Never Google images for your classes. So okay, what weakens it? Well, a lesion, quite honestly. Regularly, it shouldn't be weakened. So for those of you not familiar, I think I just downloaded this picture here. There we go. Okay, it's honestly just literally from the internet, but for those of you not familiar with where they are, okay, these are your two of your key language areas, right? I don't think I can find a better picture here. So anyway, two key areas I didn't talk about because they're not really, of course it's a motor thing, but it's more of a language. These two are more two language structures. I mean, you could have a whole lecture on language, but the general lowdown is that the Broca's area is more related to production, right? And the Wernicke's is for language understanding. And so in terms of the question of what would get it weakened, so for example, a lesion. So like not to totally confuse you, but in my lab, again, we do the visual system, but sometimes we are privileged enough to work with some patient cases with vision damage, but also because they've had a stroke with more than vision damage. So other lesions to specific parts of the brain, including, curiously, sometimes specifically just the Broca's region or specifically just the Wernicke's region. So quite in alignment with your textbook, let's say, if we find in the patient that we get matter loss or lesions in the Broca's area, then we do find that this patient really does have impaired speech. And if in the Wernicke's, you have these other patient cases with selective Wernicke's damage, and while they can produce utterances, right, you get rather impaired ability to understand language. So it's very difficult for me to sort of answer the question of what produces a weakened Broca's or Wernicke's. Like, to my knowledge, I'm not entirely sure if there could be any neurodegenerative reasons. The most common reasons I see are lesion regions, stroke regions, due to like hypoxic damage. So yeah, Jesse, that's the extent to which I can answer your question there. George, if the GP inhibits actions and releases one from some kind of pool, what are these? Oh, sorry, I didn't scroll down. Where are these different possible actions generated? Where are these possible different actions generated in the first place? I think about this. In fact, we can talk about the chicken again. I hate to always go back to the stupid chicken, but it's a great teaching tool because you guys haven't actually asked me why the chicken will still keep flopping in a coordinated manner, even without its head. Although I said, because your spinal cord or the brainstem, which might be preserved in your chicken, will still be able to send commands. Okay, so you can think of it as, I think it's one of these controversial things in motor neuroscience, where we believe, in fact, the motor system doesn't work in terms of really initiating and selecting specific fine, how do I say, like when I want to trigger a arm contraction, I don't necessarily have to systematically trigger, upper muscle, upper arm muscle, then this muscle, then this muscle, then this muscle, like this. What the current thinking is, we actually, and you might have heard me use the term motor programs. What the current thinking is, we actually have inbuilt motor programs. All it takes, and the reason why we have extensive cortical overlap, for example, from my fingers to my shoulder, is that they all belong usually to the same motor program. The same, usually all of these belong to the same motor command and motor output. And so the way to think about it is we have a repertoire of not so much individual muscle movement instructions, but we have a repertoire of motor programs. It's so hard to explain. We have a, I don't know what a good analogy is. It's much like stretching and flexing your arm is a hierarchical problem of a subseries of independent motor commands. In fact, the unit that we work with is usually the unit that we store things in the motor system. It's not so much these individual muscles and muscle instructions, but we kind of have these inbuilt patterns, program patterns, motor program patterns. And that this is actually what we are releasing when we execute an action. Not sure at all if that helped you answer your question. Great. It's so hard for it because I was gonna think of a vision analogy, but I couldn't really. Well, maybe I could. It's like much like in the visual system, you've heard that we store, we have specific neurons to look at lines and at different orientations. But when we get very higher up in cortex, we have further neurons that encode for patterns of lines that make a face, patterns of lines that, make various objects. It's sort of similar in the motor system. We encode motor patterns. Great.
I think that's a very good analogy there. Thank you, ma'am.
Yeah, it's very difficult.
May I also ask you a question?
Yeah, I don't know.
I'm really sorry for delaying our dinner time because it's also past 6 here.
Excellent.
But May I first ask, like, how, because in Parkinson's, right, you'd inhibit the motor control. Then how do the tremors come about?
Okay, so very good question. The tremors actually come from a degeneration in the midbrain. So I deliberately left it out. I deliberately left it out because it's not the, although it's visually for you guys, the hallmark of what you see in Parkinson's, it doesn't translate into degeneratively the hallmark of what is happening in terms of the neuroscience of Parkinson's. So the tremor specifically, let's address that. So it's actually due to, we currently think it's due to degeneration of a specific part of the midbrain that then leads to very similar thing that you saw in Huntington's. It leads to an inhibition problem. Okay, so that's why the tremors. So if I had taught you that, I felt like you wouldn't be able to grasp the key distinction between Huntington's and Parkinson's. But yeah, that's a very, very good question. The unfortunate part about Parkinson's. Again, let's not get too confused. The key thing about Parkinson's is the inability for the dopamine neurons to, like we just had in that first question, to where's my formula slide? Yeah, to excite the striatum in order to release action. That's the key problem in Parkinson's. But at the same time, we do have Parkinson's patients. I don't know if you're aware, not all Parkinson's patients have tremors, okay? But we do have some who have tremors, a lot who have tremors, and those tremors aren't because of this, but because of a secondary midbrain problem.
Oh, all right.
Yeah.
May I also ask about motion perception?
Motion perception, so we're talking about vision.
Yep. Do you know
Sure,
about
fire away.
do you know about the spinning dancer illusion?
Yes, I do. In fact, I don't know if you've read, did you read that from a news article?
Not really. Like, it's a kind of a meme thing.
Oh,
like a couple of years ago. But because I'm a psychology student currently, but that
right.
doesn't really address how the illusion comes about. So I'd like to ask you here.
Okay. So all those only interested in the motor system can leave.
I'm so sorry.
I'm going to see if I can find a picture, because I think a lot of people, do you have a link to the illusion? Maybe you can show the rest of your classmates.
Do I have a link? I think it needs to be a GIF.
Yeah, or like a, yeah, I guess it needs to be a GIF. Let's see. I'm so scared about what Google provides us with. Spinning dancer. So the illusion is quite simple. Actually, it's not new though. It's quite old. Are you guys aware of this? Oh well, okay, this will do. It's a stupid YouTube video. Oh my God. Okay, well, maybe you can explain. Okay, I was gonna say maybe you can explain to your classmates what the premise of this illusion is. But it's on the text here. Here, have a look.
Well, to me, it's spinning anti-clockwise.
Do we have classmates who see it spinning another way? Okay, so we get clockwise and we get counterclockwise. Okay, so are you asking about the science behind this?
Yes.
Yeah, you have to take my second year class.
Sorry.
So you're asking basically how it relates to motion perception, right? Because I think you probably had a lecture on that, did you? On how we see motion. And like, obviously I'm not going to reteach whatever you guys learned. What I want to say is actually this is not just a motion problem. This is a curious vision problem. And the way you want to understand this is when we look at, okay, look, the way we like to understand the brain is that we take sensory data and then we make a conclusion that is totally objective and totally about the sensory data. And what I want to tell you is that, in fact, we bring a lot of our own, you've probably heard this term as a psychology student, top-down information to your sensory data in order to fill in holes and in order to fill in assumptions. And the reason why some of you see it clockwise and some of you see it counterclockwise is because of some of you assuming that you are viewing this ballerina from above, and some of you assuming that you are viewing the ballerina from below. And in fact, there are other visual illusions that have the same consequence. You might have seen a rotating Necker cube. That's another example. So try when you're looking at this. Again, it's not a very long YouTube video, but try to force yourself to change your assumption about your viewpoint. And then you should be able to get her to rotate in a different direction. It's hard to get yourself to change your assumption. This is the only problem.
I focused on her static leg, I guess. I don't know, and it kind of worked.
Yeah, but this is the if we might good is sorry, I don't know how to pronounce your name, but good, exactly, that's it. So this is one of the key reasons why we have these illusions, among many other visual illusions as well. I can't find it on the internet right now, but you can see if you try to search for the rotating Necker cube, it's exactly the same thing. It's exactly the same thing. You will perceive that Necker cube to be rotating clockwise or counterclockwise, depending on what you brought into that situation. Yeah, I do kind of illusions extensively in my perception class, and the other one I'm going to send you home with, although I won't address for you in class, you can email me if you can't figure it out, is back a good 5 years now, there was the blue, black dress, white, gold dress problem.
Oh my God, are you talking about color constancy?
Yes, but that's the same premise, isn't it? Because you're seeing, your neighbor is seeing white, gold, you're seeing black, blue, based on the same physical sensory data generated by that photo, right? What's different are you guys different, you guys have different assumptions about the lighting of that scene. And that is what is driving some of you to be so adamant it is white and gold, and some of you to be so adamant that it is black and blue. So I suggest you guys go home and Google that illusion as well. But it's the same thing. So I think one of the mysteries then is about, I think I like to say, the sensory data all the time. I hammer this home in my second year class. The sensory data is definitely not the only thing that drives what you see. And very, very often, our percept is kind of altered by what we individually bring in. And unfortunately, there are many things that we can't control either. In the ballerina case, I've suggested to you that you try to force yourself to have a different viewpoint. But many of the assumptions that we apply are automatic assumptions as well, that we can't actually actively change. So it's a very curious thing, the brain.
Yeah, I think you have explained things very clearly in this
Good.
case. May I ask one more question?
I don't know, I think your classmates are dying, but go ahead.
I'm really sorry, but I want to ask about motor neurons, like mirror motor neurons.
I see.
I mean,
this is a true psychology student you have in your class.
Pardon.
Yeah,
sorry.
Mirror neurons. Yes.
So in mirror neurons, I was just thinking, because mirror neurons are those neurons that they get activated when you're performing an action, as well as when you're observing that particular action, right?
Correct.
But for observing, that would be a sensory aspect of the whole phenomenon. Mm-hmm. Then how do those neurons actually get activated?
Actually, let me bring back the slide. For those of you, in my regular class, this actually is taught, but I took it out because obviously there's just so much here. Where your classmate is talking about in terms of mirror neurons, they're actually dominantly located in the primate brain in this part of area 6 in the PMA, premotor area. And what she's talking about there is that we've got these neurons that simultaneously will respond to when I observe someone doing a particular action pattern, okay, and also when I myself am doing that action. And when I was starting my lecture, I was saying motor system is very interesting to vision scientists. And I was saying because they do share some things, this is one of the things they share. And so the question there is, how does it get activated? Right? From like if it's purely like if I'm just observing that action, but I'm not doing the action myself, and it is clearly in an area that we consider motoric, why on earth, right, would it be active? I don't want you to think about it in terms of, oh, V1 was active, and then motion area V5 was active, and then eventually finally it went to PMA and your mirror neurons. I want you to think about it like this. For some reason, okay, we have, again, this idea of having an evolutionarily built system to make us sensitive to actions. And PMA is one of these areas, much like you've probably heard of other areas around cortex that have these weird specializations, right? We have like the face area, right? And in fact, we can have a car area if you're our car expert as well. Mm-hmm. So we have an action specialization area. I think that's what you want to think PMA to be. And this cluster of mirror neurons, they're action specialized neurons. And this set of action specialized neurons doesn't really care about whether this action is triggered by visual stimulation or this action is triggered by your motoric system. The fact that you are executing this action pattern is enough to engage this particular area. And one of the key, so it's puzzling because, well, okay, given we have the visual system on its own, and we have the motor system on its own, and blah, blah, why do we need this special shared interface? You know, some people like to argue it is so that we can have a more efficient area for us to recognize other people doing those actions. And so I don't know how much you've read in terms of this area of the literature, but it has gone, like at some point 10 years ago, I turned off my literature reading switch on the mirror neurons because it got a little bit out of hand, because they believed, oh, we have this fantastic shared action area for action observation and action execution. It must be the area that is malfunctioning when we have social deficits. You know,
I heard about that.
So there's a lot of this stuff that gets kind of stretched. And I think I think we can all agree it is nice to have a specialized neural center for action recognition and action understanding, so that you can tap into your action expertise repertoire in order to understand others. So I think it's not so much we don't have the mirror neurons to facilitate our execution of actions. We have the mirror neurons to facilitate our recognition of actions.
So to recognize different actions, the mirror neuron system is there.
Yeah.
All right. Does that give us an urge to imitate actions though?
So I'm not sure if it gives you an urge. Does it facilitate? Yes.
Oh, all right.
Yeah.
All right. Thank you so much. Thank you so much. I'm sorry for delaying your dinner.
No problem. Okay, so any other motor specific questions?